Hey guys, my name is Taylor Bryden, and I'm going to be going over food microbiology, specifically wine spoilage. So first, let's go over the bacteria that performs most food fermentations, lactic acid bacteria. These bacteria are responsible for malolactic fermentation in the winemaking process, and they're found in low amounts on grapes and on contaminated equipment. Three genera that these lactic acid bacteria belong to include Oenococcus, which is the most common in MLF, or malolactic fermentation, Pediococcus, and Lactobacillus. Now, there are a bunch of other genera that they also belong to, but these are just the three that I wanted to talk about today. Some characteristics of lactic acid bacteria are that they are gram-positive, catalase negative, they are non-sporing, either cocci, cocobacilli, or rods, and they are microaerophilic, meaning that they require low levels of oxygen. So this makes sense because they can grow all throughout the wine, not just the surface. It's pretty evenly spread throughout. And it's also important to note that under certain conditions, lactic acid bacteria can cause wine spoilage. So malolactic fermentation, I'm going to refer to it as MLF, is the process in vinification where the lactic acid bacteria converts harsh tasting malic acids into softer tasting lactic acids. The overall goal of this step is to make the wine less acidic, increase its stability, flavor modification, and an overall spoilage prevention. And it's also important to mention that not all wines call for MLF. It's mostly in red wines and some white wines like Chardonnay. So this is a simplified version of MLF. You see the Oenococcus bacteria converting the malic acid into lactic acid. So this kind of gives you an idea of what it might look like. So as for the stages of vinification, these are 4 stages that I simplified for the whole winemaking process. You could break these down into even smaller stages, but for simplification reasons, I just kept it to 4 steps. So you have crushing, alcoholic fermentation, malolactic fermentation, and storage. So crushing involves destemming and juicing the grapes. It leaves you with it's called a grape must, which contains the seeds, the skin, the stems, all from the grape with the juice. It's kind of like this concoction. So during this stage, lactic acid bacteria is going to be small. So the second stage is going to be alcoholic fermentation. In this stage, yeast converts the grape must into wine. During this process, lactic acid bacteria amounts reduced, all because the yeast forms ethanol and sulfur dioxide, so it inhibits the growth of the lactic acid bacteria. The third step, MLF, is when lactic acid bacteria converts the malic acid into lactic acid and carbon dioxide. During this stage, any surviving bacteria from the previous stages is going to grow rapidly. So the final step is storage. This is where the wine is stored in a barrel and it's set to age for a given amount of time. During this stage, lactic acid bacteria amounts depend on wine composition and how it's handled. So a little recap, what's the difference between these 2 different fermentation processes? Alcoholic fermentation is when sugar gets converted to alcohol by yeast, and this happens in 100% of the wine, so all red wines, all white wines, that's how they make the alcohol. The other fermentation, it only happens in red wines and some of the white wines. So this is where malic acid gets converted to lactic acid by lactic acid bacteria. So just a little recap. Some factors that influence lactic acid bacteria growth include pH, oxygen and carbon dioxide, nutrients, vinification practices, and other organisms present. So pH is one of the key factors to controlling wine spoilage. As pH increases, the length of MLF is going to decrease, and with less time spent in MLF, it can increase the risk of spoilage. So to explain this further, a lower pH is going to inhibit bacterial survival, whereas a higher pH is going to make an environment where the bacteria can thrive and it's going to grow at a rapid pace. And that's going to increase the chance for spoilage. And an ideal pH for wine is somewhere between the range of 3.2 and 3.6. So another factor is going to be oxygen and carbon dioxide. So low levels of oxygen and a presence of carbon dioxide is going to stimulate that lactic acid bacteria growth. And this is going to go back to these bacteria being microaerophilic. They don't require oxygen to grow. That's why they grow so well throughout the entire wine. So nutrients is another factor. Yeast autolysis from alcoholic fermentation can provide energy and nutrients to these lactic acid bacteria, and that's going to ultimately stimulate the growth as well. For vinification practices, this is going to include the fruit condition and sanitizing and cleaning equipment or containers. And that's going to be one of the most important practices, because if you are using a container or specific equipment that hasn't been cleaned properly, it's going to contain any leftover bacteria from the previous use, which is going to have more bacteria in that sample. And the other factor is sulfur dioxide being added at crush to reduce sulfites and spoilage. And lastly, having other organisms present. We talked a little bit about yeast as a nutrients in the previous slide. So yeast during alcohol fermentation is going to inhibit that lactic acid bacteria growth, but the dead yeast cells after fermentation are going to act as an energy source for the lactic acid bacteria, and that's going to promote the growth. So as for wine spoilage classifications, there are obviously many different kinds of ways to classify whether or not wine has been spoiled or not. I chose these three just to give you an idea. The first one is ropiness. This is a term that's used to refer to wine that has an oily appearance. Some lactic acid bacteria can produce, it's called a dextrin slime. And when this is present, it's apparent that the wine has gone bad and it's not drinkable. The other classification is going to be the fermentation of sugars. So lactic acid bacteria metabolize sugars like glucose and fructose into lactic acid and acetic acid. And so when this happens, the resulting wine can have a vinegar-like smell, and this is due to the high amounts of volatile acids. And the last classification that we're going to go over is the other unpleasant smells. So certain compounds of lactic acid bacteria can create these unpleasant smells that can be described as mousy or geranium-like. And to go into this a little bit further, in sweet wines, specifically, sorbic acid, which is a natural organic compound used as a food preservative, is often added to the wine to prevent unwanted yeast growth. So when this is added, the sorbic acid, the lactic acid bacteria is going to attack the sorbic acid, and as a result, this compound is produced, and it gives off this geranium-like smell, and the smell makes the wine undrinkable. All of these classifications make the wine undrinkable. So let's talk grafts for a second. In this 2016 study, Cantor and their associates looked at 6 different types of red wine samples post-MLF. And they looked for the occurrence of lactic acid bacteria of the Lactobacillus genera using qPCR. So this graph here sheds light on threshold cycles. L. brevis was the first to hit their threshold cycle, you can see in the red. L. hilgardi was the next, you can see in the yellow. and L. plantarum was to follow, respectively. So we know that the least amount of cycles for detection means that there were more microbes present in that sample. So while L. brevis was the first to hit this threshold, the researchers found that L. plantarum was the most common species found out of all 6 wine samples. So after reading the rest of the research paper, I read that out of all 6 types of wine that they were testing for, L. brevis was recorded in 4 of those, L. hilgardi was recorded in 2 of those, and L. plantarum was detected in 5 of those wine samples. So to conclude, in this study, these scientists found that L. plantarum was the most common species found in that wine. And these are my references, and thank you.